guys. Hopefully you guys were able to enjoy your weekend and get a lot of studying done. Today's session won't be as long as last time, hopefully, because we only have one lecture to go over since there was no class yesterday. Also, I uploaded the notes and notes on the highlighted parts of the article that he posted. So if you guys didn't see that, I uploaded it like 7:40 this morning. So if you guys didn't see that, I'll give you a second if you want to download it to your computer or your iPad, whatever, however you guys take your notes. And then I just wanted to quickly go over all of the stuff that I have for you guys in the modules in case you haven't gone to my modules to like look at everything. So I'm going to screen share with you guys really quick. So here are my modules. So you can see that I upload each PowerPoint with my notes on it. And then I also have, like, I just uploaded this document last Friday because it seemed like we were having some trouble with homologs versus sisters. So if you want and we have extra time at the end, I can go over that. If not, you guys can just go over it on your own time. But I also have like different notes that I like uploaded for you guys. So these are all those chromatin regulating proteins and how they affect the chromatin. There's some images for you guys. And then I also wanted to just point out that I do these important things to know documents. So when I took the course, our TA just had like a massive document. He just typed all his notes and he had like a 45-page document, and that's what he would upload for us. So these are my notes of what he had highlighted, what I thought was important, and I just rewrote them out for you guys and took out some of his fluff and condensed it to 7 pages for the first 3 lectures. And then I also uploaded one for lecture 4, or the second half of the lecture 3, if that's how you want to call it. Yeah, so definitely take a look at those because it has some important stuff on there. I just didn't want to upload his like 45-page document and freak you guys out because a lot of it was fluff. And then I also have these helpful videos for you guys. So I don't know if you've like actually gone in here and clicked on some of these videos, but they're pretty good. So if you're struggling with a topic or you just want to like watch to see if you understand a topic, this is pretty helpful. Okay. So with that being said, we will jump into it. Unless anybody has a question. I downloaded the app to my phone and I have the chat open to my phone, so hopefully that works. So if you guys want to type something in the chat, hopefully I'll be able to see it. Okay, so he left off on this slide last time, so it's kind of just like a refresher slide. So we have DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon. So we know that DNA polymerase alpha works after the primase. It's the initial polymerase that starts after primase. So it initiates replication and of the lagging and the leading strand. Then we have DNA polymerase delta. And here it just says Okazaki fragments elongation, but he verbally said also the leading strand. So make sure you write that in there. And then DNA polymerase delta, they used to think that it was only like in the leading strand. That was the main synthesis for the leading strand, But then they did that experiment where they found out that it was just redundant, and if they removed DNA polymerase epsilon, DNA polymerase delta would just take over. So he wants us to deprioritize that. Again, it's going to be a decoy on the test. Don't get tricked by that. Yeah. Oh, so here we can also see that it's not circled on this slide. It'll be on this, like on the next 2 or 3 slides. This exonuclease, 3' exonuclease activity. So polymerase delta and polymerase epsilon have this 3' exonuclease activity, whereas polymerase alpha doesn't. And he goes into what that means here. So it's a corrective mechanism, and it's done by delta and epsilon, as stated on the previous slide. So sometimes we'll get this weird form of a nucleotide, and it has a proton on its R group, and that allows it to like base pair to the wrong base pair, not Watson-Crick base pairing. So sometimes normal Watson-Crick base pairing is interfered with by these weird nucleotides with their protons on their R groups, and we get this mix match. We know that A is supposed to go with the T and C is supposed to go with the G, so we have this weird thing happening here. But it's so unfavorable that it will quickly disassociate. And then now we're just left with this weird short single strand of DNA that's exposed. So he said in lecture it will be more than just one nucleotide, but this is just simplified for us while we're learning the concept. So now we have this short single strand of DNA that's exposed. So our DNA polymerase will recognize that short single strand that's exposed. And it's going to want to cut that out. Like, that's wrong, we can't have that, we need to cut that out. So if we know that DNA polymerase synthesizes from 5' to 3', and we know we're always adding new nucleotides to our free 3' hydroxyl, here we can see that that's our free 3' hydroxyl, right? So if we're going to go back and cut it out, it's going to proceed from 3' to the 5', so opposite of the way we synthesize. So it's going to chew that back from 3' to 5' until it gets back to double-stranded DNA. Once it gets back to double-stranded DNA, it's like, okay, I excised what needed to be excised. The rest is good. And now we can continue on with our normal synthesis or replication. So on this figure, it kind of just shows us more, okay, now it's disassociated. Now we have our single strand of DNA that's exposed. We're not going to be able to add on more nucleotides to that single strand that's exposed. So then DNA polymerase will chew that back from the 3' to the 5', and then we're left with our double-stranded DNA that matched correctly. So we have the correct Watson-Crick base pairing here, and then now we can finally go ahead and synthesize or replicate more of our DNA. And this figure, he said not to worry about it. It's just if you want to understand how that weird matchup between the incorrect Watson-Crick base pairs can occur, but he does not care about the chemistry aspect. So here you can just see how a T is able to bind to a G and a C is able to bind to an A. But we don't need to memorize these structures, and we don't really need to know like why or how that's happening. He just wanted to show you in case it helped you understand this concept of having a C go to a T. So then here we have that chart again, and now he has it circled after he went through it, the exonuclease activity, 3' exonuclease. So just keep in mind though that the exonuclease activity is dependent on strand separation. So if we don't have this single strand of DNA that's exposed, it's not going to do it. It's not going to work. So it's dependent on that strand separation. Okay, this slide, I don't really remember there being a question on our exam on this slide. the main takeaway for this slide is just that we're always going to have errors when we replicate our DNA. So when we start to synthesize our DNA, we'll have 1 in 10 to the 5. When we do our proofreading, you know, we correct 99% of the errors. We still have 1 in 10 to the 2. Then we have this other mechanism that we're going to learn more in depth later, this mismatch repair. We have 1 in 10 to the 3. And then when we combine all of those, it's 1 in 10 to the 10. So every cell division, we're going to have some error. That's the main takeaway. Although we have mechanisms for proofreading and checking for errors, we still are going to have some, which makes sense because that's how we can get cancer after, you know, once you're older, after however many cell replications, something will go wrong with one of these errors, and that's how you can develop cancer and stuff. Okay, this slide, I'm sorry, my handwriting is kind of messy and it looks kind of crazy, but it's not bad. So basically, up until this point in this PowerPoint, we've been talking about DNA replication, but we've kind of just been looking at DNA by itself, and we haven't even touched on what happens to all those histones that we learned about in lecture 2. So now he's kind of tying it in together for you guys. So the nucleosomes will come apart partially, but not totally. They're not going to totally disassociate from the DNA. They're only going to come apart partially, but they do that so they can make a space for that replication machinery, and we can get in there and replicate that DNA. But specifically, histone genes don't have any nucleosomes. And that kind of makes sense because it's easier for us to just transcribe those genes if they're already available and they're not tied up in those nucleosomes. So it's more efficient because if you think about it, we need to have a lot of histones. When we're synthesizing new DNA, we need to like synthesize histones pronto for that new DNA that we're synthesizing. So remember, we have a histone for every 150 base pairs. Well, there's like 3 billion base pairs in your DNA. So hopefully that illustrates it for you, just how much we need histones. So there are 500 copies of histone genes because we need a lot. So we need lots of histones for each new DNA molecule. That's why we have 500 copies. And again, they're not bound up in those nucleosomes, and they're synthesized in G1 so that we can prepare for S phase when we're replicating our DNA. So now that we've figured out, okay, we're not completely disassociating our nucleosomes when we're replicating our DNA, we don't have histones or nucleosomes on our histone genes, and we have 500 copies of those genes. We're going to synthesize those histones in G1. Okay, now that we've done all of that, how are we going to get them on our new strand? Well, that's why we have this NAP1 protein. It's a chaperone protein that facilitates the reassembly of nucleosomes. So N is for nucleosome, A is for assembly, and P is for protein. So kind of easy to remember. So it's just going to load those nucleosomes onto that newly synthesized DNA. Okay, here's our summary of proteins to know. So we know helicase, we know topoisomerase, DNA polymerase delta, primase, DNA polymerase alpha, RNase H, DNA ligase, pCNA. Don't worry about this clamp, it's pCNA. And then what we just learned about now is our nucleosome assembly protein. So there's supposed to be a T there. It's our anchoring point for remembering histones and nucleosomes need to be dealt with for DNA replication. And these are all potential right answers on your exam. And then again, DNA polymerase epsilon is a decoy on the exam. Yeah, so just don't get tricked by that. And what you guys should be able to do is like make a list of all of these proteins and just write down what they do so that you don't get confused on the exam. maybe make like a Quizlet and make like flashcards of these, because that's something that you can make really easy. Or you could just have your study group quiz you on them. Okay, we've seen this slide a few times. So we talked about our replication origin. Now we're going to go into our telomeres. That's it. But first, he wants to talk to you about hybridization. So this is kind of similar to what we learned during lecture 1 when we talked about probing that DNA. So you could take like 1,000 base pairs and label it with like fluorescence, and then denature the DNA that you want to tag. Does NAP1 resemble nucleosomes or G1 or G2? You know, I'm not entirely sure about that. I would say during S phase, because if you're replicating all that DNA, you need to be able to coil it and wrap it so that we can have enough space. And then we know that during mitosis, they're all the way condensed. So I would say during S phase, we're synthesizing that DNA and then already wrapping it around those nucleosomes, but I'm not entirely sure. That's a good question. Maybe you can email him that and have some interaction with the professor. So, okay. You're welcome. Sorry I couldn't give you like a definitive answer. Again, guys, I don't have a PhD in this stuff. I just did well enough in the course to kind of offer you my insight. Okay, so back to this idea of hybridization. So we can take like 100 or 1,000 or however many base pairs we want, and we can label it with fluorescence. So this one has a yellow fluorescence, this one has like a light blue fluorescence, and then we can denature the DNA that we want our probe to target. So we denature that DNA to allow our probe to Watson and Crick base pair with it. So we mix the probe and the DNA, and then that probe finds its complementary sequence, which is the DNA that we wanted to target, and then they Watson and Crick base pair. So in this image, it might look like we only have 2 chromosomes here. So we have 1 homolog from your mom and 1 homolog from your dad, but now we can see that we've targeted 2 copies of each gene. So that means we have 2 sister chromatids in each of these homologs, which makes sense because we know that we need to use metaphase chromosomes whenever we're taking a karyotype. So does anyone have any questions on this? Do we understand the homologs versus sister chromatids? Okay. So if you get a picture like this on the exam and he asks you how many sister chromatids are here, you're going to say 4, because there's 1, 2, 3, 4. Because he will put an image on your exam and he will ask you how many sister chromatids or how many homologs are present in this image. So you need to be able to differentiate. Okay, so now we're going to talk about telomeres. So what I wanted to do before looking at these slides is I found a video, it's like 2 minutes, for us to watch. Oh wait, if I screen share with you guys, you can't hear the sound, can you? Okay, then I guess what I'll just do is that I'll send you guys this video for you guys to watch. Ugh, I wish we were in person. I'll send you guys this video for you guys to watch after we're done talking about it, and we'll just look at this depiction, I guess. Is there a share computer sound option? You know, I don't know. I'm not a tech whiz, guys. We're all just doing the best that we can. Let me see. It's share screen. Yeah, I mean, it's okay. I'll show you guys this this like depiction, this image with its caption, and then you guys can watch the video. The video is just kind of reiterating what I'm gonna read you right here. And then we'll go into it on the slides, and then you can watch the video if you still want to see like the animation with the words over it. But okay, so our chromosome.
Do you find Khan Academy helpful?
Yes, I do. I'm pretty sure I linked some Khan Academy videos, or maybe I don't know. I think there might be one or two Khan Academy videos in my helpful videos document that I uploaded for you guys. If you are wanting to find more information or you need to see it animated to understand, I would try Khan Academy. I also like the Amoeba Sisters. Yeah, I would just recommend those two. Those are pretty good for going over these topics.
Okay. Thank you, Ivy.
Uh-huh. Okay, no, I think I need to screen share again. Okay. So we know that our chromosomes are linear, right? So that's why we run into this problem where we need telomeres. So DNA at the very end of our chromosome cannot be fully copied in each round of replication, resulting in a slow, gradual shortening of the chromosome, which is bad because then if our chromosomes are shortening, obviously we're going to lose important information in our DNA with each replication. We think about how many replications of DNA each cell goes through, and like that's a lot. That's bad. Then we'll lose function of those cells because they won't have all the necessary information. So basically, you know, we have our leading strand and our lagging strand, which is called Okazaki fragments when we synthesize them. And we know that it begins with an RNA primer every time because DNA polymerase cannot self-initiate. So in most cases, the primers of the Okazaki fragments can be easily replaced with DNA, and the fragments connected to form unbroken strand, we know, by ligase. When the replication fork reaches the end of the chromosome, however, in many species, including humans, a short stretch of DNA does not get covered by an Okazaki fragment. Essentially, there's no way to get the fragment started because the primer would fall beyond the chromosome end. So there's not enough space for us to add in our primer, and we know that's a problem because DNA polymerase can't self-initiate. So if we don't have a primer and DNA polymerase can't self-initiate, that region of the chromosome is just not going to be replicated. And then also we run into the problem of this primer on the last Okazaki fragment that does get made can't be replaced with DNA like other primers. And we know that RNA does not transmit the information, it doesn't carry the information from the parent cell to the daughter cell. So then we're still going to run out of our coding regions of our DNA. So that's why we get telomeres. So they're going to just add in this junk DNA to allow for space for that last primer and then for DNA polymerase to come in and synthesize the rest of that DNA. So here's our replication fork. And then if we look down here, after replication, okay, we see that we don't have enough space here for our primer and then for DNA polymerase to synthesize this on both ends. So then we're going to just basically cut out this portion of DNA, which is not good. So then now, if we look at our slides, also keep in mind that primase is an RNA polymerase, whereas telomerase is a DNA polymerase. So now if we look at our slides, I wrote in here, telomerase adds junk DNA so that the primer has plenty of space to make Okazaki fragments, and when it comes time to lose that primer, we don't lose important DNA. So here we have an RNA primer. this region right here is not going to get synthesized or replicated because we don't have space for another primer over here to be laid down and then DNA polymerase to come in. So that's why we have telomerase adding this junk DNA over here. You can see now that this chromosome is longer than this chromosome. So then now we have space for our primer to be placed and our DNA polymerase to go ahead and synthesize more DNA so we can actually replicate this portion of the chromosome that we were missing. And we know that all of this over here is just junk, it's not coding. So it doesn't really matter if this piece over here gets cut off. So that's going to protect our chromosomes from the loss of important DNA, and it's also going to protect our chromosomes from exonuclease activity. Because if we had just these single strands, DNA polymerase might come in and cut it back until we get double-stranded DNA again. So telomerase has its own template. That's how it's able to synthesize these junk DNA sequences. And it's an RNA template inside of this polypeptide. So this blue right here is its RNA template inside of this polypeptide. So one end of the template looks like the other end of the template. And that's how it's able to come up, match over here. Okay, here's my template. And then I'll synthesize some new DNA. So it's an RNA template that synthesizes DNA. And then you can see here, okay, now this is the new sequence that we added. And then that gives primase enough space to lay down a primer. And then that gives DNA polymerase enough space to go ahead and synthesize the rest of that strand. And then, you know, we can replace the primer that we had previously with DNA. So I'll send out that video. I'll just add it to a helpful videos tab under my modules so that you guys could go ahead and watch it in case you want to just see the illustration. But I think that Khan Academy did a good job of explaining it for you guys. And if you have any more questions about it, you know, ask me right now or email me. But I think it's pretty understandable. We just have an RNA template in this telomerase, and we're synthesizing DNA based off of that RNA template to save the end of the chromosome. Okay, now we're getting into this pathway. So does anyone have any questions before we get into this? You can type it in the chat because I'm seeing your chat on my phone.
Ivy, can I ask over here because I can't type in the chat for some reason?
Yeah, no worries.
In regards to, oh my gosh, I just blanked. Sorry, I'll ask again later.
Yeah, no worries. So, okay. This might look a little complicated, but we'll break it down. So let's first talk about this E2F. So E2F is a transactivator. And if we remember what transactivators are, I could go back. Oh wait, no, that's lecture 2. You can't go back. Transactivators, if you guys don't remember, will recruit a hat. And if you remember what a hat does, we'll add acetyl groups to those histones and remove its positive charge. So that way it's not associating so tightly with DNA. So we will loosen our chromatin and make our DNA accessible. So that's E2F. It acts as a transactivator. It recognizes one of the regulatory sequences in the histone genes or DNA polymerase genes. So it has to recognize that sequence and bind to that sequence. And then it will bind to that sequence and it will recruit that hat so that we can have access to those genes that are needed for S phase. Okay. Then, so that's our E2F. So we want E2F if we want to replicate. So then RB is going to inhibit E2F. So if we go to this slide first really quick. here's our E2F, this white little dotted thing, and here's our E2F site. So it bound to its site, the specific sequence of nucleotides that interact with E2F. E2F will not interact without this. And that's different than what happens with our histones because histones have no specificity. So E2F will not interact without this. So then we have RB, and we said up here that RB acts to inhibit this E2F. So RB binds to this HDAC, and if you remember what HDAC does, it removes those acetyl groups on the histones so that the histones can regain their positive charge and interact with negatively charged DNA, so then we get tightly condensed chromatin and our DNA is not accessible. So RB is bound to an HDAC, making sure there's no acetyl groups. So then if we can't access the genes that we need for S phase, we're not going to replicate our DNA, we're not going to make new daughter cells. So RB binds to an HDAC, and its job is to inhibit this E2F. So now let's make it a little more complicated, as he would say. So RB, if it's heavily phosphorylated, then it will not interact with E2F. But if it's lightly phosphorylated, then it will interact with E2F. So if we want to replicate and we want to make new daughter cells, then we will heavily phosphorylate this RB, so it's not able to inhibit this E2F, and that means this E2F will be free to bring in a hat and leave those genes like open. So if RB is heavily phosphorylated, will not interact. Lightly phosphorylated, it will interact. And RB stands for retinoblastoma protein because it was first found in retinoblastoma, but it's present actually in a lot of cancers. So when we want to replicate, an outside signal will remove RB and its HDAC, which will leave E2F free to bring in the hat and access the genes needed for S phase. But now, if RB is defective, which is common in cancer, regardless of if it's phosphorylated or not, the complex will not work. And E2F will always stimulate, that's what this little S stands for, will always stimulate transcription of S phase genes. And that's why we get cancer, because we're always replicating, we're always creating new cells. Does anyone have any questions on that? Okay. Can you go over again what makes this more specific? Yeah, so it has a sequence-specific site that it needs to bind to. So if our cell gets some outside signal, he said that we're not going to go in depth on what that signal is right now. But if our cell gets some outside signal, then E2F can come in, bind to its specific site. RB will be heavily phosphorylated, so it's not going to inhibit this E2F. And then E2F will act as a transactivator. It will bring in a hat and then make those genes accessible for replication and transcription and so on. So it makes it different from histones because they have no specificity. E2F needs a sequence-specific site on that DNA to bind. Does that make sense? Do we have RB in all cells? I'm pretty sure we do. Is E2F only found on some chromosomes or genes or in all? So I'm pretty sure this is happening in all of your cells because although we term RB retinoblastoma protein, it's not exclusive to retinoblastoma. That's just the first place they found it. So I'm pretty sure that this is happening in all of your cells because we need this E2F if we want to replicate our DNA and go through mitosis. So we need this E2F to bring in that hat and allow those genes to be accessible. but we don't necessarily want to replicate our DNA and divide all the time, or else that's cancer. So that's why we need this RB to inhibit this E2F. So I'm pretty sure that this is found in all cells. Might not be happening at all times. There might be other mechanisms at work. It's definitely more complicated than what he's showing us right now. But yes, we can think of it like that. You're welcome. Okay. So then now he wants us to revisit PCR. So lightly phosphorylated RB equals activation of transcription, right? Yes. No. Not lightly phosphorylated. Heavily phosphorylated RB. Because if it's heavily phosphorylated, then it will not interact with E2F, leaving E2F free to be a transactivator and bring in that hat. So if we want transcription, then we want a heavily phosphorylated RB and E2F is active, bringing in a hat. If we don't want transcription, then RB will be lightly phosphorylated and it will inhibit E2F, and there will be RB will be bound to an HDAC. So now for PCR. So if you guys remember PCR, we have our strand that we want amplified. So then we get primers for that strand. We denature that target strand so that it's accessible to the primers. And then we DNA polymerase and synthesize new strands. Okay. Well, he wants us to keep in mind the polarity. So if we look at all the way to the left, we can see at the top we have 5' to 3', and at the bottom we have 3' on the left and 5' on the right. So you have to keep that in mind in order of which way we synthesize, which is 5' to 3'. So if we look at this bottom strand all the way to the right, okay, we said that that was 3' to 5'. That means our primer will bind over here on the left and synthesize from 5' to 3'. So that means it has to be opposite on the top strand because we know that they are anti-parallel. So just keep in mind the orientation of how our DNA polymerase is working here. And then now he added this new concept of instead of heating up that DNA to denature it and then cooling it down so that it can re-anneal and doing that over and over and over again, instead of doing that, we can just add a helicase. So it will unzip that DNA a little bit, allow our primer to bind, and then allow our DNA polymerase to synthesize from that primer, which cuts out the time of heating it up and cooling it down and heating it up and cooling it down. So then here's just another example of how many cycles we can do. So we gave like a good example of an exam question here. Which of the following 3 items is required for PCR, or which of the following is not required for PCR? So if we look back here, well, first we need DNA that we want to be amplified. Then we need primers for that DNA. Then if we're not going to be heating and cooling, we need helicase. Then we need DNA polymerase, and we need those nucleotides. Because if we don't have nucleotides for DNA polymerase, then it's not going to have anything to make that new strand of DNA with. So we need nucleotides, we need DNA polymerase, we need helicase, we need primers, and we need our DNA that we want amplified. And then on this image, he shows us the fluorescence with the number of cycles. So on this red strand, we started off with not a lot of double-stranded DNA. That's what this DS in front of the DNA is. Okay, hold on, pause. It's a tiny amount of double-stranded DNA, but that's why we have low fluorescence to start off. But then once we do PCR, we end up with a lot of fluorescence up here. So the number of cycles of PCR has increased, and now we have a lot. Now compare that to this blue line. So if we compare that to the blue line, we see that it takes us more cycles to reach the same point. So if it takes us more cycles to reach the same point, we started off with even less DNA here than we did over here. So it took us more cycles. We needed to replicate even more to get to the same level of fluorescence. Does that make sense, guys? Okay, if there's no questions, I'm just gonna keep going. So here are your fill in the blanks. I filled them in for you guys to check your work, but still try to be doing them in the discussion board so that he can answer you guys. So now, if there's no questions, I will pull up the highlighted article that you guys had to go over.
Hey, Ivy.
Hey, yeah, you have a question?
Yeah, for the polarity on the PCR slide, so were you saying that I don't want to say it wrong, was the primer the one going 5' to 3 and then the new strand was doing Okazaki, or am I overthinking it?
So you're gonna have, so on both strands, you're gonna have both occurring, the leading and the lagging strand. So if I go back to here, this is what a replication fork looks like. So on one strand, you're gonna have Okazaki fragments and you're gonna have a leading strand, and on the other, you're gonna have the same Okazaki fragments and a leading strand. So you're gonna need primers for both. For the leading strand, you need to place a primer first, and then DNA polymerase will take over and will just continue on straight until it reaches an endpoint. But then for the Okazaki fragments, you're gonna need primers every time. So just depending on how much DNA you want replicated, and depending on the image that he's showing you. So if we look all the way to the right on this bottom one, we see that this is the leading strand because it's 5' to 3'. So then if we look at this top strand, it's still the leading strand. It's 5' to 3', but it's just in the opposite orientation than the bottom, because we know that they're anti-parallel. So in this image, he's just showing you both of the leading strands, and he just wants you to know that they're going in basically the opposite direction, because, you know, we have anti-parallel polarity. And just remember that if this is the 3' all the way to the right on this top strand, then we know it's the 5' that needs to be like with it. Does that make sense?
Yeah, thank you.
Uh-huh.
Evie,
Do
can
we-
you please go over E2F, HDAC, and like the relationship with RB?
Yes. Okay. So E2F is a transactivator. So that means that it's going to recruit a hat so that we can add acetyl groups to the histones and we can open up that DNA so that we're allowing for transcription, we're allowing for replication. So that means all of those genes needed for S phase, E2F is bringing in a hat and allowing for those to be transcribed, and we're allowing for our DNA to be replicated. RB serves to inhibit E2F because we don't want transcription of S phase genes all the time, or else our cells would be replicating all the time. And that's what happens in cancer. So we need RB to inhibit E2F until we need to actually replicate our cells. So RB inhibits E2F. It does this by recruiting an HDAC. So the HDAC is going to remove all those acetyl groups on the histones, and then it will condense that chromatin. So those genes needed for S phase can't be transcribed, and we can't replicate our DNA. That RB will only inhibit E2F if it is lightly phosphorylated. So if RB is heavily phosphorylated, it will not inhibit E2F, and that's when E2F is free to become a transactivator and recruit a HAPT.
Thank you.
Mm-hmm. So we don't need RNase-H for PCR because we're using a DNA primer. Should we put the fill in the blank in the exam material discussion thread or make a separate discussion thread for each fill in the blank? I'm not sure. When I was in the course, there was just a discussion section for fill in the blanks. Let me check this out for you guys. Yeah, see, fill in the blank for lecture 3, fill in the blank for lecture 2, fill in the blank for lecture 1. So I would just put them there. Yeah, discussion posts are not for a grade. They're just for a studying tool. And at least when I was in the program, he went through and answered people. I don't see that he answered, but he does go through and answer people. All right. So if there are no more questions, then what I will do is pull up okay. So he gives you these articles that he wants you to read the highlights only. And sometimes you might be reading it like, okay, how am I supposed to understand just like this highlight if I don't understand the whole sentence? But you don't actually have to read this whole thing. I never read the entire articles. I only read the highlights, and sometimes if I needed some more context, I would read the sentences around the highlights just to understand why he highlighted that one word in the sentence. And then I also took some notes. I uploaded this for you guys, so we'll go over my notes after. But basically, since we learned about telomeres in this lecture, he brought up this article where they found how telomeres are related to certain diseases. So telomere dysfunction has been suggested in degenerative disorders like idiopathic pulmonary fibrosis, and it's a short telomere. So we know what telomeres are, right? DNA protein structures that protect chromosome ends from degradation. And they found out that discarin is an essential component of the telomerase enzyme. So there's like a group of disorders that they term the telomere syndromes. So here we go. We're talking about the discarin. It's the best characterized mammalian telomerase accessory component. So telomere elongation is cell cycle regulated. So telomerase elongates telomeric DNA by adding telomere repeats during S phase. So you should know that it's during S phase. So We need to understand how long telomeres are supposed to be to understand if we have a shortened telomere and that's what's contributing to our disease. So they did that and they figured out that it's 92 telomeres in a human interface cell on average. So here are some of the diseases. Familial IFP is just this idiopathic pulmonary fibrosis. And then we talk about this dyskeratosis congenita. It's classically defined as mucocutaneous features like skin hyperpigmentation, oral leukoplakia, and nail dystrophy. And the primary cause of mortality are bone marrow failure, pulmonary fibrosis, and cancer. So dyskeratosis congenita is characterized by the short telomere defect. Sometimes they've seen it de novo. And all of those mutations are for encoded telomerase or telomere protein components. So we know that there's something wrong with our telomeres there. I think that's the last highlight. Yeah. So then we see that adult onset manifestations of telomere-mediated disease are related to germline TERT or TR loss of function mutations. And the frequency of these mutations is highest in individuals with IPF. So then he highlights this image, a long telomere, we form normal B and T lymphocytes, a short telomere, we get bone marrow failure. Okay, so now if we look at the notes I took. So telomere dysfunction, if we have shortened telomeres, has been found in some degenerative disorders. Our typical manifestations are idiopathic pulmonary fibrosis and bone marrow failure. Telomeres are DNA protein structures that protect chromosome ends from degradation and fusion. They're cell cycle regulated. Telomere repeats only added during S phase, and it's an average of 92 telomeres in an interphase cell. We know that discarin is an essential component of the telomerase enzyme, which is making those telomeres. It's the best characterized telomerase accessory component. So then dyskeratosis congenita is linked to mutant telomere gene characterized by the short telomere defect. These are what it's classically defined as. Then that's the primary cause of mortality. And then we've seen some de novo cases. So the TERT gene is a gene that provides instructions for making a component of the telomerase enzyme, and the mutation or loss leads to telomerase-mediated diseases. And the frequency of those mutations is highest in individuals with the progressive idiopathic pulmonary fibrosis disorder. And then here are just the images that he highlighted. So that's how I would approach these highlighted portions. Sometimes you have to read like a little bit around to understand the context, but don't waste your time and read the whole article. Unless you enjoyed it and you want to read it, then go for it. But if not, just read those highlights and make like a little note like this. And he will pull questions from these highlights on the exam. So like, for example, he could ask you like, during what phase are telomeres added? Then you have to say S phase. He could say, what's an essential component of the telomerase enzyme? And then it would be discarin. So don't just blow off these highlighted readings. He will pull like one question from each reading, probably. Does anyone have questions on the highlighted reading? Okay, so if there are no questions, then I have some questions for you guys. So if you guys want to just type your answers like what we did last time in the chat. Okay. Exonucleolytic correction by DNA polymerase proceeds in what direction? Nice, you got it. It's from 3' to 5'.
Hey, Ivy, I have a question.
Uh-huh.
I saw that in the chat someone.
Oh no, you cut out.
Hello? Hi. Can you hear me?
I can hear you now. Oh, what's going on? No, I can't hear you.
I keep getting muted.
I don't understand. I haven't muted anyone.
Hi. I'm trying to ask a question, but I keep getting muted. I don't know why.
Yeah.
Okay. So in the chat, someone's saying, do we need RNase H for PCR? And I was
No,
oh, okay, we don't?
no, because we're using DNA primers.
Oh, okay. Okay, thank
Yeah.
you.
Mm-hmm. Okay. So true or false? The nucleosomes come apart completely. For replication, that is. Good. False. They don't. Which description fits telomerase? Yeah, so that's going to be an RNA templated DNA synthesis. So it has that RNA template inside, but it's using that to synthesize new DNA, what we call junk DNA. When RB is active, it is bound to what? Yeah, so when RB is active, we want to inhibit that E2F. So we're going to bind an HDAC and remove all those acetyl groups from those histones to make sure that we are condensing that chromatin. We're not allowing those genes to be transcribed for S phase. In PCR, what can be substituted for heat to access the DNA for replication? Good. It's helicase. So we unzip that DNA a little bit, allow for the primers and DNA polymerase to access that DNA. The primer will bind, it leaves that free 3' hydroxyl for DNA polymerase to then synthesize the rest of the segment that we're looking to amplify. All right, and that is it. I will stick around if anybody has any questions for me. It was kind of short because we only had one lecture, but that's good. More time for you guys to study.
Hi, Ivy. I didn't really understand what you meant by RNA templated DNA synthesis for telomerase.
Yeah, okay. So let me screen share with you to show you what I'm talking about.
Sorry if you explained it like 1,000,006 ways, but
No, that's okay. All right. So you enter full screen. Okay. So telomerase has this RNA template. So it's using this RNA template to synthesize new DNA. So that's why I termed it an RNA templated DNA synthesis. So up here we said it's a DNA polymerase because we're synthesizing new DNA nucleotides, but it's using an RNA primer to do this. So it has its own RNA template, its own intrinsic template, so that we can synthesize DNA. Does that make sense?
Yeah, so like telomerase just kind of sits on the template strand with like RNA nucleotides in it, and then like polymerase just goes over telomerase and makes the.
No, so it can do, so it has its own RNA template, but it
Okay.
acts as a polymerase. So it uses that template to synthesize DNA segments.
Okay,
Yeah.
that
Okay.
makes sense. Thanks.
Okay, good. Can you repeat what's necessary things for PCR? In lecture, he said there were 3 things required, but we went through more than 3. Oh, he was just giving an example of like what 3 options could be required. So then it would be like nucleotides, DNA polymerase, helicase, and a primer. So that's 4. But he can say, that's why I think he corrected himself and said which is not required, because he can put DNA nucleotides, DNA polymerase, helicase, and then he could put like topoisomerase or something. And then you would have to pick, okay, which one is not, and that's topoisomerase. Or he could say, yeah, I think that's how he would term that question. It's more than 3 things. He was just thinking off the top of his head. Yeah, no problem.
Did you post your notes on the articles, or is that just an example?
No, I posted them, and I will continue to post them for you guys, but I was just showing you that that's how you can kind of approach those articles for yourself. But I have it posted in my modules. Yeah, it's right there under lectures 3 and 4, part 2.
Hi, Ivy. You explained this so many times, but you said that when RB is heavily phosphorylated, there's no binding with E2F, and then that's what activates transcription?
Yes. So
Okay.
E2F will recruit a hat. So that will add acetyl groups onto those histones. Those histones will lose their positive charge. And if they lose their positive charge, they're not as closely interacted with the negatively charged DNA. So that allows it to open up a little bit and decondense so that that DNA can be accessible for transcription. How many exam questions based on the articles do you think we'll get? Like 1. Probably. Because I don't remember exactly how many highlighted articles we had to read for this first exam, maybe like 2 or 3, but he's not going to waste like 10 questions on those articles.
Quick question, Ivy. Can you go back over the histone genes and I guess like how gene replication occurs within like those regions?
Are you talking about this slide, or are you talking about from lecture?
Yeah, this slide.
Okay. So when we're replicating our DNA, our nucleosomes will not completely disassociate from that DNA. They'll only partially come apart to make space for that replication machinery. So he gave the example of this H3, H4 tetramer still bound to that DNA. So it's not completely coming apart. So some are still being left on that DNA. And when we're replicating our new DNA, we're going to need to make a lot of histones. So the histone genes don't have nucleosomes themselves, so that we can transcribe those genes like way more efficiently. We don't have to partially break down that nucleosome to allow for access. They're just accessible. And we have like 500 copies of those histone genes so that we can make all the histones that we need, because we have histones every 150 base pairs, and our genome is like 3 billion base pairs. So if you can do the math, that's a lot of histones that we need. And they're synthesized in G1 because we replicate our DNA in S phase. And then the proteins that facilitate the reassembly of the nucleosome are chaperone proteins that are termed NAP1. So nucleosome assembly protein 1.
So NAP1 reassembles those nucleosomes that, or the histones that were broken apart from those nucleosomes.
And it loads it onto the new strand.
Gotcha.
Hi, Ivy. I remember my question now.
Oh, perfect.
In regards to telomerase, I was always like told that it was only in germline cells. Do you know anything about that?
I'm pretty sure that is false.
Okay. That's why I wanted to confirm with you guys.
Yeah.
And then for helicase, this is more of a curiosity question, if you don't mind me asking. But I'm interested in like the helicase used for PCR. Like, how do you make sure that the helicase is basically opening up the DNA and denaturing it in front of where you want the primer to go? Like, is there a specific sequence that it has to open up to? Because I know it usually opens at the origin of replication.
Yeah, so I'm not necessarily sure about that, but it can just open up the entire DNA, but that primer won't bind until it finds its complementary sequence.
Yeah.
So it might not even be specific to open up right where that sequence is. It might just open up the entire strand of DNA slowly, and then that primer only binds to its complementary sequence. Or they might have honestly found a way to alter that helicase, but I don't know. I'm sorry.
Okay. All right, thank you.
Does anyone have any more questions?
I have a question, Ivy. So I saw in your notes that telomerase prevents fusion of the chromosomes. I was wondering why that would happen.
So if you have one single strand that didn't get replicated, it might fuse to another single-stranded chromosome that also didn't get replicated, and then you'll have fusion of 2 chromosomes and create like a whole nother chromosome, basically. Does that make sense?
Yeah, thanks.
I had a question, but it wasn't entirely related directly to genetics, just as a disclaimer for anybody else who's on the call who may want to leave or something. I feel as though like we keep getting closer to the exams, and I'm just wondering, like, how did you compartmentalize like all this information? Like, how often did you go back to like lecture 1 and lecture 2 to study it for the exam?
So I would go back to it like on the weekends. So I would go through my week and I would go to lecture, rewatch the lecture and make sure I got done all my notes. I would go to the TA sessions. So basically after going to lecture, rewatching the lecture, and then going to the TA sessions that week, I would hear that lecture like 3 times. And I would take my notes on it, and then I would just continue on with the week like that. And then once I got to the weekend, that's when I would meet with my study group, and we would kind of just run through all of the lectures for that week in like different blocks. So we'd break it up a little bit. We'd go through, someone would read one part, the other would read another part, then we would quiz each other. So I would go back to that week's material and the week before on the weekends. I would start off on like a Friday morning or Saturday morning, just reading over the old material before I got into the new material that week that I wanted to like really sit with.
Okay, and then did you do that for each of your classes, or did you find that you had to give more time to certain classes than others?
I found that I did have to give more time to certain classes than others. Personally, genetics, I didn't have to put that much time into, because once I went to lecture and then rewatched the lecture, I kind of understood it, but I had a background in genetics from undergrad. So like anatomy, for example, I had taken anatomy in undergrad, but not to the level that you guys are taking it now. So I would have to spend more time in anatomy. So you basically kind of just figure out, especially when you do the practice questions that we give you, the weekly Q&As that we give you on Friday, then you can kind of figure out what your weak points are and try to like focus on those and bring those up to speed, if that makes sense.
Okay, yeah, thanks.
Mm-hmm. Mm-hmm. Since you guys that are still here, do you want me to go through that sister chromatids versus homologs document, or do you want to just keep asking me questions? So to make sure. Sure, go through it. Okay. Let me answer this one question in the chat first. So to make sure the telomerase extends the DNA by adding repeats of about 92 nucleotides in order to extend room for the DNA polymerase to synthesize from the primer, and it will incidentally also add protection to the DNA ends. Yes, that's correct. So when we replicate our DNA, there's always going to be an end that doesn't get replicated because there's not enough space for the primer, right? So that's technically like a single strand of DNA. So if you remember when I discussed the exonuclease activity, that subunit in DNA polymerase looks for a single strand. So if you have a single strand, it will just get excised by that exonuclease activity of DNA polymerase. So it protects our chromosomes from the loss of important DNA because we added that junk DNA to make space for that primer, so that primer was able to enter, I guess you could say, and then the DNA polymerase can synthesize from there. And then whatever is not replicated from that junk DNA will get excised or will get destroyed. But that doesn't matter because it was just junk. Okay, hopefully that answered your question.
Ivy, are you going to keep posting those lecture summaries? It really helps.
My important things to know.
Yeah, they really help. It's awesome.
Yeah, of course. Yeah, that was just my version of condensing my TA's crazy document.
Hi, Ivy. I have a question.
Uh-huh.
Can you explain one more time about why we are going to find the junk DNA on the new DNA strand?
It's not getting added to the new DNA strand. It's getting added to the template DNA strand. Where is that? Am I still screen sharing? Can somebody let me know if you can still see my screen?
Yes.
Okay. So that junk DNA is getting added to this orange, which is the template strand. So the junk DNA is elongating the template strand to allow for more space for that primer to be laid down. So if we're like looking at this top chromosome right here, there's not enough space for a new primer to come in. So telomerase adds that junk DNA to the template strand so that there's more template for the primer to come in, lay down it, primase to come in, lay down its primer, and then DNA polymerase can build off of that, which is what we're seeing down here. We have our primer, and then DNA polymerase is synthesizing there, and now this last primer that was laid down can get replaced with DNA.
Thank you.
Okay. So now I can go through this document. Is that big enough for you guys to see? Okay, hopefully it is. What's happening? If anyone wants to ask me a question before I go through this, just stop me. Okay, so humans have 23 pairs of homologous chromosomes, right? You get pairs, plural. So you get 46 individual chromosomes, and 23 come from your mom and 23 come from your dad, making up 46 individual chromosomes. The homologous chromosomes are determined by similar length, centromere position, and genes. So they're not exactly the same, but it's just like if, for example, I had a gene that determines eye color on this chromosome from my mom, then the homologous chromosome that I get from my dad will also have that gene that determines eye color. And that's why we group them together, because they're similar, but they're not exactly the same. So one of the homologs comes from your mom and one comes from your dad, so they're not exactly alike. When we're preparing for mitosis, these homologous chromosomes will each replicate themselves and form an exact copy termed sister chromatids. So when we're preparing for mitosis, we need to have a copy of that chromosome from your mom and your dad for the new daughter cell that we're making. So that's why they will exactly duplicate themselves. And that's what we term those sister chromatids. So these sister chromatids will separate in anaphase and retain their own centromere. Each daughter cell will then have 23 pairs of chromosomes. So if we look at this image here, we can say like purple comes from your mom and green comes from your dad. So they're similar chromosomes, but not exactly alike. Like for example, my mom has blue eyes and my dad has brown eyes. So this one can be from my mom and this one is from my dad, but they both have that gene for eye color on this chromosome. So that's why they're homologous. So now we want to replicate our cell. Okay, we need to make an exact copy of each chromosome so the daughter cell can have the right amount of chromosomes. So these exact copies are sister chromatids. So if you look to the left, we have one homologous chromosome, two homologous chromosomes. And then if we look at the right, we still have one homologous chromosome, two homologous chromosomes, but now we have 1, 2, 3, 4 sister chromatids. And we term them chromatid because they're still attached at this centromere. So now if you look at this image, we can see, okay, we have two chromosomes, one from mom, one from dad. Now we're going to prepare for mitosis. So we're going to replicate ourselves. Now we're lining up and then we're getting pulled apart. And now each daughter cell has a copy. So we're back to one chromosome for each. So 2 chromatid pairs, 2 times 2 is 4 chromatids. That's what this is saying up here. There's 2 chromatid pairs. So pair means 2. So there's 2 pairs, that's 4 chromatids. So now if we look at a karyotype, homologous chromosomes are paired up, then they are put in order from largest chromosome pair to the smallest. So one from your dad, one from your mom, one from your dad, one from your mom. But you can see that they look similar, but that doesn't mean that they have the exact same genes. And we know when we're taking a karyotype, we have to use a metaphase chromosome right here. So that means that there are 2 sister chromatids in all of these homologous chromosomes. So chromatid is a term we use when there are 2 exact copies of a chromosome still attached at the centromere. So here's a homologous pair, one from your dad, one from your mom. They're going to get replicated, and they're still attached at the centromere. In this image at the top, there are 2 homologous chromosomes right here, or 1 pair of homologous chromosomes. In the image at the bottom, there are 4 sister chromatids, or 2 pairs of sister chromatids. And there are still 2 homologous chromosomes, or 1 pair of homologous chromosomes. So hopefully that kind of clears things up with that. Does anyone have any more questions for me?
I have a quick question.
Mm-hmm.
So if you have 1 chromosome that's replicated and you have 2 sister chromatids, you would technically have 2 DNA molecules, right? Not 1?
Correct.
Okay. Thanks.
Mm-hmm. Well, if that's all, then feel free to email me if you guys have any more questions. And I'll see you guys this Friday and next Tuesday for another session. All right, you're welcome. Bye, guys.